Okay, we're gonna go ahead and start. So welcome everyone. We're extremely excited to have Dr. Yang Liu from University of Illinois Urbana-Champaign. She's a professor of electrical and computer engineering and also bioengineering, but she's also the deputy director for NIBIB funded Center for Labor-Free Imaging and Multiscale Biophotonics, also known as CLIME. It's a very nice acronym. Before joining the University of Illinois, she was a professor at University of Pittsburgh. She got her PhD degree from Northwestern University, and her lab has been developing wide range of optical microscope techniques such as labor-free, quantitative phase microscope, super resolution fluorescence microscope, and computational imaging. So we are very excited to see what's been up to with Dr. Liu's research. So welcome. Let's welcome us.
Thank you. Thank you so much for having me here. I really enjoy my visit. This is such a unique place. Very few places have that scale of optical sciences as here. So my lab has, I think here is optical science. I love talking about optics. So I'm a microscopist. So we develop optical microscopes to address various problems with the main application in cancer. I think we all know the importance of light microscope, both in basic research as well as the clinical applications. So if you look at lots of the biological or clinical problem in general, it's really from an imaging standpoint, it's a multi-scale problem. So every scale is important from, for example, in case of tumor, early cancer detection, from detecting tumors at the microscopic scale, we're looking at cell-cell interactions and tissue microenvironment at the mesoscale, and looking at sort of morphological changes of nuclear architecture at the microscopic scale, or understanding some of the molecular scale changes at the nanoscale. So every single scale is very important. And with advances in light microscope, so one of our goal is really to develop sort of an all-in-one system that connecting all different kinds of length scales, everything done on the same piece of tissue that we can get. The same multi-scale problem goes to the temporal scale. Lots of the fast dynamics happen at the millisecond scale, and some of the disease development really take months or years to develop. So again, light microscope could have a very important role capturing these dynamic changes over time as well. So this is sort of many years of my research, we've been working on sort of at the molecular scale, and more recently we sort of expanded to mesoscale. So the goal, I think the future is really having a super powerful light microscope that can capture all the way from macroscopic to nanoscopic, and also a wide range of temporal scales. So my lab sort of have several general directions. We work both on quantitative phase imaging and high throughput super resolution microscopy. And more recently, we start working on sort of a cross-scale, multi-scale that connecting both sort of a label-free refractive index changes of the tissue with molecular scale information that you get from super resolution imaging. Many of my research really motivated by the goal to understand how normal cells or normal tissue transform into cancer. So we know this is really a multi-step process through 20, 30 years of accumulation in most human cancers. So that's really a long window of opportunity for us to capture cancer before they become malignant. So each of these stages, normal transform to a small tumor or to a larger tumor, eventually to invasive cancer. And the gold standard is actually a physical property, right? If you look at these changes, there are morphological changes of either architecture of the tissue or the nuclear morphology. So we spent many years understanding what are the nuclear morphologies, can be physically detected using optical technique. And this is sort of just list, it's not a complete list, but it's showing some of the characteristics in the nucleus, like the size, shape, chromatin texture. One of the amazing things that I always, as I learn more, I always appreciate is we talk about, these days, talk about mutations and lots of molecular changes, but this kind of physical change is actually independent. It's almost ubiquitous across different tumor type. So that's, from engineering standpoint, that's a good news. But we know this kind of microscopic changes lots of time is not sensitive enough, especially at the personalized level. If we want to figure out which patient likely respond to a specific treatment or which precursors had higher risk for developing cancer, those are the areas that precision medicine will come in. So that's when we start talking about molecular changes. So if you look at the things, all the molecules inside the nucleus, we know they're DNAs, but DNA are not randomly organized in the nucleus. They're actually highly compartmentalized. So the DNA is wrapped around these proteins called histone proteins. They form these nucleosomes, and they further compacted into either open or condensed form. So this physical compaction of the chromatin actually very important regulating how gene gets expressed. So that's why it's important in cancer. Because a sort of abnormal gene expression is an important hallmark of cancer. But when we start talking about molecular changes, it's a completely different language. So molecular biologists typically use biochemistry approach. They don't really think in terms of the physical characteristics. So even now, I think there's very little we know how this molecular compaction contribute to the changes we see clinically in the histology slides. So if I push, this is sort of a question I had, it's probably when I first started my position, independent position 15, 16 years ago. So if I push a little further, looking at, from a technology standpoint, can we design better technology that can capture this molecular scale chromatin structures that can detect cancer early, or predicting sort of the therapeutic response using the higher level of sensitivity that we have. So that's where I got interested in super resolution microscopy.
That's where the chromatin.
So these are the individual nucleus. These are what you see, the dense packed structure there. Those are chromatin. The nucleosome, this individual level, typically you won't see it under microscopic level. I'll show you some examples later. You will see at the super resolution level, you can see some group of nucleosomes that compact. So the histone size, I think the typical size is about less than 10 nanometers. Is 5. I think in general it's about 5 nanometer, but typically they compacted. So when they folded, it's about 20, 30 nanometer. That's the size of the nucleosome. Yeah. So the approach we are using is called single molecule localization based super resolution. Just for the students in the audience, I think many of you probably are already familiar with this approach. We know light microscope is diffraction limited. So if you have a point source, you don't get infinitely small spot. The size of this spot is defined by the diffraction. But for years, people know the center of this can be localized at a much higher position. So in the traditional diffraction limited image, because you have 4 force labeled across the entire structure, so you don't really get those single molecules. So with some help in photochemistry and photophysics, we actually can switch most of the molecule into the dark state, and then leaving a small subset of the molecules on, and then that creates this single molecule or sparse condition, and then we can localize the center of each and eventually reconstruct the super resolved image. So this is sort of what you see in action. So you basically just like a blinking stars and each of the frame you get, and then we accumulate this because the random is a stochastic localization. You just accumulate sampling. So eventually we can get a reconstructed image that's about 10 times better resolution compared to the conventional diffraction limited. So routine resolution, we get roughly 20, 30 nanometers. People are pushing down to below 10 if your photon is super bright. So, and another reason there are different forms of super resolution microscopes, you probably know. The reason we choose a single molecule base, also the instrumentation wise, it's a very, very simple system. It's just a wide field microscope and plus sort of a drift correction system that you maintain the stability of the focal plane throughout the entire data acquisition. But I started this about 10 years ago. So when I first doing it, there are many limitations. The images were beautiful. You get those from bacteria or the cell cultures, people show those very nice images. But to apply this to the problem I want to study, which is look at how normal cells turn into cancer in the tissue context, it's much harder than I expected. So we thought, how we can really bring the power of super resolution to advance our understanding of cancer in a more clinically relevant setting. So it really took us many years to address many technical issues, including the throughput, robustness, which means lots of time you put the sample there, different type of sample, you may get different resolution because various technical issues. And when it comes to clinical samples, no one even tried it because most people in this field came from biophysics and they'd like to work with a really ideal model system. And another issue is they don't really know what to look at. What is really cancer relevant? So I want to go through some of the steps, but that's really kind of 10 years of work. One issue is we know the illumination, when you use laser for wide field illumination, is a Gaussian shape. But for single molecule localization, it's very critical to have a more uniform illumination or power density. Otherwise, the blinking rate is different. So to address that, most of the time we find a compromise. We just take the center part. It's still not completely uniform, but it's more or less. So that really limit the field of view. So in recent years, there are different versions of flat field illumination. So you can use beam shaper to shape a Gaussian beam into a flat field. And the device, it's expensive lens. So the approach we use is actually we use a multimode fiber with a vibration piezo vibrator that creates a really nice wide field illumination. The upside of this is you actually don't, so this is the performance, actually a very nice 95% uniformity. And the nice thing is we don't even have to use expensive laser. So even those industry level laser diodes, lots of times it works. And one of the most challenging part of the instrument is the drift. So you have to maintain the nanometer stability throughout the entire process. So this is early days, we used to use markers to correct for sample drift. Basically, we place a marker either on the cover slip surface or on the sample. And because they inherently have this variation, they're not going to be uniformly located on the same depth. So we use this variation actually to locate, to jointly estimate the depth, and then use it to track it every a couple of 100 frames, and then correct it, do online corrections throughout the process. So the precision here, we got roughly 5 nanometers in the Z direction. So this works. We use it for, actually before I moved to Illinois, we use it for like almost 10 years. But it's a pain. So when you move, you have to take your instrument apart. And I thought, I don't want to repeat the same thing anymore. I want to build something better. So I didn't want to use the marker. So the approach, we actually got inspired by the auto-focusing mechanism that people use in photography. So we basically start using these LEDs with two oblique illumination. If they're autofocus, there's a lateral shift. So this is actually the mechanism that photography autofocusing use, and the creating this sort of the phase shift in the Fourier space. When we try this in the super resolution system, it turned out it's actually a really nice linear dependence with a little bit of sort of this is we suspect it's because of the some issue imperfect alignment around the focal plane, right around the focal plane. But overall, it's a nice dependence over a large range of the focus. And when we look at the precision, it's actually even better than the marker base we had before. It's sub-nanometer. So this is sort of a newer system we are using now. It works very nicely. We use that routinely now in the lab. And this is sort of the image we get. It's very robust. It doesn't matter the index matching, the density of the cells. It really creates a very nice, stable 3D drift correction. So this is an example of microtubules. And after the drift correction, we get you really need that drift correction system to achieve resolution approaching 20, 30 nanometer. This is an example of 3D. And you can see they maintain the Z focus as well. So with high resolution, that's a pretty typical. You get slightly worse resolution in the axial direction, but that's something you can accomplish with this very simple approach that kind of borrowed the idea from microscopic imaging. Another issue is the background. So ideally you want, because you want to find the center of those individual emitters. So you want very bright emitters, very low uniform background. But in tissue, you don't get that. You get all this kind of variation. And this actually well recognized even 10 years ago. The field recognized if you have a little bit of background that skew your localization, so they can introduce inaccuracy up to tens of nanometers. So in case of resolution at that scale, that's a big deal. So to overcome, so this is really one of the biggest challenge for tissue imaging. It took us almost 5 years to get to the point that we reported this so-called path norm, basically a super resolution imaging system adapted for pathology samples. So if you use a standard paraffin embedded tissue section, and that's what you get, you use very high laser power density, you end up getting lots of background. You don't see the single molecule. So we did a little bit of clearing. This is actually a very simple clearing we do with the index matching buffer. And because of reduction in the spherical aberration and the scattering, we significantly lower the background. So now you can see those individual molecules. So it's about 80% of the background reduction. And this step is fundamentally lowering the background, but it's not 100%. So we had to use sort of because this is a temporal sequence. So we can estimate the background, which is relatively stable across time, versus the blinking molecules, so they have temporal variation. So we use that property to estimate the background and remove it from the original raw data. And after this step, we get a much better improved resolution as well as reduced image artifact. So that's a critical step for us to get reproducible data. So this is one aspect in terms of improving the robustness, but there's also throughput issue I talk about. So this approach, single molecule localization, inherently you need the sparsity of those individual molecules that's spatially sparse, and then we localize the center. So overlapping molecule is something bad because you basically lower your resolution. So we typically reject them. The problem is then you have a limited sampling each frame. So it requires standard 10, 20,000 frames to accumulate enough sampling to reconstruct the image. So essentially, this approach sacrifices the temporal resolution to gain spatial resolution. So one of the easy thing to increase the throughput is you make it denser, right? So you kind of go somewhere in between this ultra dense versus the sparse molecule. So you see some overlapping molecule. And if you increase the density by a factor of 10, you can reduce accumulated frames by a factor of 10, which means you reduce your acquisition time by tenfold. So if you can still reconstruct the same image, that will be ideal. But the problem is it's harder. It's mathematically, it's much harder to do. So it's fairly slow image reconstruction. So when we first looking into this problem, we thought, the traditional deconvolution will do a pretty good job. And we actually tried. It turned out it's true. If you use Richardson-Lucy deconvolution, it works. It can separate those overlapping emitters fairly well. But it's very slow. If you apply this to tens of millions of molecules, those time will add up. It's just unrealistic. So we thought, what if we use a linear deconvolution? Because it's non-iterative, it's much better. But the problem is noise. Noise is really a big factor. But the earlier work I just talked about, we figured out a way to remove the background. So it turned out that really helped making more stable or more better performance linear deconvolution. So we basically, the first step, we removed the background, and then we put everything in the Fourier space. This is just a linear deconvolution, the inverse filtering, and we truncate the high spatial frequency noise, and then put it back to this image space. So now you get a much better separated fluorescence emitters. So that's basically a method we use to expedite our image reconstruction. Together with a GPU acceleration, we can reconstruct image in real time. So which means the image reconstruction time is shorter than the image acquisition time, and still getting a decent resolution. So when we have all these factors combined, we can really have a high throughput super resolution imaging system. So this example looking at DNA origami and the 3D super resolution imaging and also tissue. So this is a field of view is roughly 200 microns. At if I zoom in, it's like more like a Google Earth, right? So you kind of can zoom in and look at molecular scale changes. So this resolution is about 30 nanometer. So that's sort of the first part of my talk, talking about we develop a series of robust, high throughput single molecule localization toolkit to enable high speed image reconstruction or image acquisition for single molecule localization, from flat field illumination to drift-free development, and optical clearing to lower the background, and also the high speed image reconstruction. And if you look at my list, there's one part we have not addressed is what is the molecular changes there. So let's get to the chromatin studies that we looked at. Essentially, we are labeling condensed region of the chromatin. So we are using this antibody-based labeling, targeting histone proteins that's specific, that's enriched in the condensed region of the chromatin. So we use this animal model. This is a very common intestinal carcinogenesis model. So, and we look at 2 time points. Normal, which is a sort of 6 weeks very early. There's a mutation there, which means the animal eventually will develop cancer. But this is the phase that's they're still without the tumor being formed. And the second phase, we look at there's some formation of the tumor. So this is the data we had. So this red color here, this is the microscopic wide field diffraction limited. This is a super resolution. So you see this individual one is a single nucleus, and the dense packed regions are those condensed form of chromatin. So to answer the question, what is the size of the chromosome, the nucleosome? So we really have to zoom in to that specific region. So you see those microscopic foci, now you see those individual clusters, and that's basically a group of nucleosomes. And And I will tell you exact size in the next slide. So, but basically what you see here is actually quite to our surprise. We expect some changes between the normal versus tumor, but the early stage, you see the chromatin, the compaction level of the chromatin already disrupted, so they become more loose. And this continuous process throughout the entire carcinogenesis. So when we quantify, it's basically we either call it nano domains formed by heterochromatin, or some people call it clusters. So if we do this clustering method, we can measure the size of these. So the size of this nano domain on average, the smaller ones go to around 15 nanometers. But that's really the group of nucleosomes. It's not a single nucleosome. The larger compact ones, because I don't think we can really differentiate the single. So it's really a group we measure at the, so normal all the way to the tumor, you see gradual decompaction throughout the process. This is just, we want to get a little more unbiased analysis. We did correlation analysis instead of the clustering. They show you the same thing. It's a shorter correlation length as the carcinogenesis develop. So this is sort of an antibody-based labeling. And every time you do fluorescence, biologists always ask you a bunch of questions, and you have to cross-validate. So we did this. They also want us to look at DNA. So this is the work that kind of motivated by the reviewer's question. We start looking at, so we actually had to make our own dye that conjugate Cy5, which is the one that typically used for single molecule localization with the host that delivered to the nucleus. And then that enable us to look at, to do super resolution on the labeled DNA. So what we see here is normal pre-cancer and cancer. These are patient samples. So you see similar to what we found earlier. The nucleus in the normal case, especially at the periphery, there's a very dense packing. When they get to the precancers and cancer, you see gradual disruption. And when they get to really this tumor of invasive cancer, you see more of a diffuse pattern. That's basically the disruptive pattern kind of migrate inside. And if you look at the sort of the conventional, it's kind of seeing the pattern, but that you need that resolution to really identify the actual size. So this is sort of the measurement we did on the DNAs with the size roughly 100, 120 nanometers of the DNA nanodomains. So moving forward, I'm not a chromatin biologist, so this is sort of a nice discovery. We did lots of validation on the biological side. I'm just showing you a small portion, but I'm more interested in how we actually applied it in the clinical setting. So one of the applications we are exploring is to basically re-stratify colon polyps. So there are, once people go for colonoscopy, up to 50% of those patients end up with some sort of polyps. And the surgeon, the gastroenterologist will remove them, but they always suspect how soon I need to bring the patient back, what are their risks. So we want to address that clinical need, unmet need. So here is an example looking at non-advanced adenoma, which is sort of a smaller tumor. This is advanced adenoma, but without high-grade dysplasia. So these are larger tumors, but pathologically they're very, very similar. And when we measure the chromatin compaction level, what we saw is those advanced adenoma, they have more disrupted structure, which means they're further along in the carcinogenesis process and at the high risk. So similar, another example looking at patient with Lynch syndrome. So these are basically patients with germline mutations. Their DNA cannot repair. As a result, they have a higher risk for developing colon cancer. And we looked at all of the patient tissue we look at are normal. And the group affected Lynch, these are the patients that previously developed cancer, the tumor removed. By the time we look at their tissue, these are all normal. But if you compare those normal tissue versus the true healthy controls, you clearly see more fragmented chromatin compaction compared to this densely packed structure from the normal patient. So what this tells you is sort of this concept of field cancerization. The cancer affects surrounding tissues that we essentially, this is another angle to justify this compaction chromatin could be used as a sort of a risk stratification marker. So that's what we looked at. Healthy control versus affected lynch, each of these dots average about 200 nuclei per patient. And unaffected lynch, these are the patients with mutations, but haven't developed cancer yet. So we see sort of a spread. And one of the hypotheses is this could be, those ones closer to the cancer could be the higher risk patients, but those will require follow-up. So far, I talk a lot about sort of developing this high throughput super resolution imaging platform to understand how chromatin changes at a microscopic or nanoscopic scale. So this development allow us to expand, so from roughly 20 nanometer all the way to roughly 200 nanometer. So that's 4 orders of magnitude. But this is only part of the cancer biology or the cancer development you can look at the smaller, too small to see. But there are also areas that are too big to see. So in recent years, especially the importance of immunotherapy, people start to recognize cells don't work alone. They work by groups. So the cell-cell interaction is very important. So that's kind of expanded to the mesoscale. We want to look at cell-cell interactions, how they work as a group, how they work, why some cell type close to the other, like how who you want to be friends with. So it's the same for cell. So we want to also understand that level of the spectrum in cancer. So with that motivation, we want to develop sort of a mesoscale system that can capture as many cells as possible while maintaining that high level resolution. And ideally, we want to link all the mesoscopic changes all the way down to the nanoscopic. So we can get both molecular scale information together with the tissue microenvironment all on the same tissue. So that's kind of the goal, but we know traditionally there's a limited trade-off between the field of view and resolution. You can get a large field of view, but you compromise, you settle with lower resolution. You can get high resolution, but you limited, because high NA lens you use, you limited the resolution here. So to compromise, so the traditional way is you have to scan and stitch all the images together. So that's okay if you are just imaging a small number of samples, but the limitation is if you repeat this for hundreds of samples, that's going to be very time consuming. The second thing is when you scan it, you sacrifice your temporal resolution. So you cannot really capture the large cell dynamics. So that's very important if we want to understand how cells respond to a drug treatment. We want to capture all of those cells at the same time. So this is more recent work that we developed this multi-model, multi-scale mesoscope. So the paper get accepted. It's actually coming out in 2 weeks in Science Advances. So we basically sort of leverage the bigger lens. I saw a lot of bigger lens here in this institute in astronomy. So it's actually, we did borrow some of the hardwares. The cameras we use is actually an astronomy camera. It's a consumer level astronomy camera. It has very large sensor, small pixel size, coupled with it's a bigger lens that people use for stereoscope. So when we couple with the right sampling and pixel size, the NA here is 0.5. It's actually one of those highest NAs we can find off the shelf. And we build this system with the goal of getting as large of a field of view as possible with the highest possible resolution. And in this case, we combine the flat field illumination with sort of a bright field that's used for phase imaging, and then the multi-channel fluorescence. So this is basically a single field of view. We get roughly 2 by 3 millimeters. You know, one of the issues, I think many of you here probably know this, the field is not that flat. So there's definitely a field of curvature aberration. Even everything is focused because these lens are not really designed for high resolution microscopy. So you see a lot of aberrations at the corner of those. So that's also reflected in the actual image we see. So to compensate that, we actually use computational-based corrections. A very simple, straightforward way. We just divide those into multiple regions, and then we do a deconvolution using our calibrated point spread function. So it definitely improves the overall performance. So this is the final result we get. 700 nanometer within the center, slightly worse when you get to the periphery, but it's overall sub-micron resolution. Actual resolution is still not ideal because you know the NA square dependence. When you lower your NA, that definitely compromise the actual resolution. So I'll show you some examples toward the end. We try to overcome this limitation. So again, we want to do phase imaging, just basically taking a stack of images and do a transport of intensity equation-based phase retrieval that can retrieve the phase over a large field of view. So these allow us to capture, this is a single field of view, roughly 3,000 cells, and you can still get sub-cellular level details. So this is, you can see the cytoskeleton here, and we monitor them dynamically. So you see the dynamic changes. This is mitosis that we capture in this process. So you can do a lot of the standard things we typically do using quantitative phase imaging, like monitor the drug response treated with the chemotherapy drug. You see a slowdown of the dry mass over time. And for the fluorescence imaging, we also validated the resolution. So this is the nuclear lamina, the nuclear membrane protein. This is centromere, the center of the center of the chromosome. So if you look at the resolution here, we validate the width of this spot is roughly 700 nanometer. So that give you, so that's sort of validated in biological cells. So one of the really nice thing about this system is they allow us to detect rare events that wouldn't be possible if you just capture a small field of view. So one of the things we looked at is to look at drug-resistant cancer cells. And this is a single field of view image. And then when we reviewed those videos, we found this very interesting effect. So if you look at this video here, you see the nuclei over here, it seems they get engulfed or sort of swollen by these larger cells, and it's not done yet. So you see, they kind of, each frame is 2.5 minutes. So it's actually pretty fast, they start to divide. So, and we see that very often in the drug-resistant cells, not in the typical cancer cell. So this kind of rare event is interesting, but the thing is, a lot of times you see an effect that looks interesting, but what does that mean biologically? So what we did, the next step is to do a high content phenotyping on the exactly same set of samples. We basically monitor them dynamically and then fix them and do a sort of a cyclic fluorescence imaging with multiple colors. So this set only showing here is 12, but we did, I think, a total of 30. So here just showing different molecular markers that reflect this dynamic behavior. So it turned out these polyploid cells, these are the cells that actually one of the mechanisms they use to survive the drug treatment and develop chromosome instability. So we found these cells have very high level of stemness. So CD44 is a stem cell marker, a very high level of stemness. So you see another It's intentionally shown this because this is another multi-nuclei or polyploid cells. They all have very high level of stemness and high level of DNA damage. That's probably why they get sort of hidden inside as a protection of themselves, and high level of metabolism and proliferation. So they sort of just capture lots of the molecular characteristics that help us interpret those dynamic changes that you can capture, so that you get the full picture of what is happening underneath those morphological changes. So to further link the fluorescence microscopy, as I said, this mesoscale get down to 700 nanometer, but not really super resolution. So to cross that scale, we actually use image registration. So we can kind of find that same cells with the same sort of linking that to the super resolution image that acquired using a different system. So this is really a one-to-one correspondence. So it allows us to sort of looking at all the population level changes, the phenotype, high content phenotype, and then linking that to the molecular level changes. So here is just example showing the red is chromatin and the green is the nuclear membrane. So you see that the spatial separation between these two. So to further understand this problem of how the cancer cell develop resistance over the course of treatment. So we compare the two populations. One is the control. One is the cells that survive the treatment and eventually grow back again. And if we look at the super resolution level, we see this chromatin actually gets more condensed as more compacted as a way to develop resistance or escape the treatment. And if you look at the periphery of the nucleus, we also see a more dense patterns, at the nuclear periphery. So that's indicating the traced molecular characteristics they develop as a response to the drug treatment. And then we compare, so for the super resolution data, because we can link back to the high content molecular data, so we can really compare, look at their phenotype, combining phase information together with multiple molecular markers to understand their molecular phenotype. So for example, this is a control cell, so you can see the proliferation level overall is still higher. These are the cells that recover from the treatment. So their proliferation level not up yet, but you can see their DNA overall is getting smaller. When they divide, somehow they have to lower their DNAs as a way. It's one of the signs for chromosome instability, and their overall lower level proliferation, more condensed chromatin, as I showed earlier. So if we look at many cells, they sort of summarize those characteristics of drug-resistant cells. Morphologically, their dry mass, which is the parameter you measure from phase, is lower. That's consistent with overall less DNA content, but nuclear size is similar. Proliferation level, because these cells still recovering from those, lethal drug treatment, they're not that highly proliferating yet. And chromatin-wise, they become more compacted as a way to escape the treatment and then condense the nuclear membrane. All this sort of partially could be pointing out eventually they're basically trying to lower the level of DNA damage to reduce their susceptibility to the lethal drug. So that sort of just give you an example of how we can combine this multi-scale information to gain insight for cancer, how the cancer cell develop resistance. And the same platform, you can use it for sort of multi-model imaging for whole slide tissue. So this is the example we are currently exploring, combining standard HAE, quantitative phase, and fluorescence. So you can see this is a normal region, it's an inflamed region. You can see different architecture, and these are different immune cells in comparison with epithelial cells. So that really this multi-model approach allow you to see a lot more than you would typically basically get with a single modality. So this is some example looking at control, inflamed, and remission. So the goal here is we want to be able to predict, sort of giving an assessment of the molecular scale wound healing process. So eventually we can predict whether those patients in remission who are likely to develop recurrence down the line. So this is some examples doing this multicolor fluorescent staining. We did a computational analysis. Here, just counting the percentage of cells, different cell types. So you can see, for example, the inflamed cells, that's clearly immune cell type more predominant. And in remission is somewhere in between. So this is sort of a way to quantify different cell types. And then for further classification, we combine the phase. So these are the phase values that we get from the tissue. So you see inflamed tissue, the chromatin is actually, this is just from the cell nucleus. The refractive index or the phase become lower. So that indicates more open, less dense chromatin compared to those ones that either in remission or in the normal control. So these are just different fluorescent markers that indicate sort of the ones in remission. It's between the inflamed state and the healthy controls. This is it's a immune cell activation marker. So we are able to sort of quantify this among different cell types. So eventually, this is a principal component analysis. We show the classification of different patients. So this is still, we want to add to more of a spatial interactions. And with this data still ongoing, it's a fairly small patient size, sample size. So the next step, as I mentioned earlier, we want to go with 3D. The problem with the low NA mesoscope is, the axial resolution is not good. So if we look at the thicker tissue samples, you see lots of blurring effect because all the signals are added up. And what we use here is sort of a clearing plus expansion. So expansion is a physical process. So you basically embed the tissue into a hydrogel, and then when you add water, they kind of expand uniformly. So lots of groups in the brain imaging field, they use this approach. So this is a combination of clearing and also increase the resolution. So essentially, you physically expand the sample to achieve super resolution. So when we try that approach, and this is what you get, it's a 3D. So you can see this individual cell nuclei. You can even see the sub-cellular structures, and the color-coded is the depth information. So you definitely get three-dimensional information with this approach. So this platform is, this is more like a close-up view of the 3D information. So this is really sort of an expansion plus mesoscope. It's a really nice platform, because when you physically expand the sample getting so large, it's much harder to image using conventional microscope. But mesoscope, because they can image large area in a short period of time. So our ultimate goal is to do this high throughput 3D volumetric super resolution. So you can get sort of really multi-scale from nanoscale, because this is super resolution, all the way up to mesoscale. There's some of the ongoing work. We sort of really want to expand the super resolution further to have a whole slide super resolution imaging platform. And lots of component has been addressed. So now it's really the automation part to make it robust to scan the whole slide. And lots of the ongoing studies, we want to expand using this mesoscale phase imaging platform to look at how we can quantify this large scale dynamics of cell morphology in the drug screening context, how that assess the drug response. And then again, leveraging the throughput from mesoscope for 3D spatial biology. And one of the last component, we really want to push it to face imaging at high resolution. So I don't have, so it's still pretty early. I just want to show you one example. When you look at phase at high resolution, the information is truly amazing. So I used to look at phase from tissue at a low resolution level. I thought the contrast is never sufficient to differentiate anything. But now you look at, this is the normal tissue, it's the inflamed tissue. You clearly see these are the vessels, these are different immune cells. You can even see fiber structure from the cytoskeleton and some separations of the cell membranes. And it's just so much rich information you can see. This is the inflamed tissue. You can see the granular structures from the eosinophil. You see different type of immune cells. So one of the ongoing work, we are doing sort of a virtual staining, training the AI algorithm correlate with the fluorescence and see if we can use this sort of morphology to differentiate different cell types in this context. So I want to take a few minutes to also advertise our center. So this is part of the reason I moved to Illinois. So our center CLIMB is label-free imaging and multiscale biophotonics. So it's one of the 3 optical imaging centers that are funded by NIBIB. So the goal is really to establish as a technology and training hub to address important challenges in biomedicine, either enable basic biological discoveries or clinical application. I think some of you probably know the story behind the center. So the center was initiated by Gabby Papascu, who is really was one of the pioneers in quantitative phase imaging. He unfortunately passed away just a few days before the center award was announced. So after that, Steve Bopart stepped up to become the director of the center. So they recruited me to serve as a deputy director. So basically my role is to foster the collaboration from both inside and outside the campus. So any of you, I know there's a large body of, people in biomedical optics. Anyone interested in label-free or multiscale imaging, we really welcome collaboration. So now it's the 3rd year of the center. So we are working on the renewal 2 years later. So we're really looking for new partners for the next phase of the center. So I want also advertise a couple of opportunities. So this is symposium we are hosting next year on April 10th. It's going to be at the Beckman Institute. And we welcome really the greater biophotonics community to participate. If any of you have questions, feel free to reach out to me or Steve. So these are this Rohit Bhargava, he's the expert in infrared imaging. Mark Anisasio is the expert in AI. So it's a really strong team that we are welcome, collaboration from people around the country. And then one last thing is the summer school. So just like your winter school, so we have sort of, as part of the CLIMB educational component, we host the annual summer school. So that's the last week of May is 1 week. And the targeted audience are basically trainees, graduate students and postdocs, even research scientists. So we cover all the accommodation and food. So you only need to travel there. It's a 1 week program. And we've been hosting this for actually, even before CLIMB, we have this for many years. So the topic mainly covers label-free imaging and nonlinear optics and computational imaging. So there are actually lots of hands-on labs and also data analysis. We also have industry partners, like this year we have people from NVIDIA, GSK, they come and talk about their platform. So, we welcome applications. So so we do select roughly 20 people, around the country. Actually, include some people from Europe to join us. So keep that in mind. Welcome application of our summer school. Just at the end, these are my group. I moved to Illinois in January, so a lot of the data I presented come from this group. I have a Pitt, and now I have a new group with many more students. A couple of people move with me, but these are really the people that did all the work I presented. All my clinical collaborators, those are still ongoing with my colleagues at Pittsburgh. So thank you all for your attention. So here are my contact. Feel free to reach out to me if you have any questions.
Any questions? And for those of you who are attending the talk, via Zoom, you can type in question and I can read the questions. So who's going to start off?
Thank you, Dr. Lin. My name is Kevin Figueroa. I'm from the Dr. Brady Array Camera Lab. My research is in the multiscale challenge.
Oh, cool.
I was wondering if I could ask you.
Sure. Yeah, absolutely.
When you guys talked about doing fusing the data from the low NA and high NA, we're taught here that convolutions are shift invariant, but they aren't scale invariant. So I was curious how you were forming the registration of multiple scales.
Oh, we have to scale it. Like, first there's a scaling factor, and then we just use cross correlation to find a location.
Oh, so you scale the wide field of view?
Yeah. We have to, I think the super resolution, we have to scale it down.
Right.
Yeah.
So it's memory intensive.
It is, yeah, it is memory. Yeah.
Thank you.
Because the super resolution image is 20,000 by 20,000.
Yeah.
Yeah.
That's why I was like, that's pretty big.
Yeah, it is.
Yeah. What computer are you using?
It's standard PC with 64 gigabytes of memory.
Oh, so how long does it take to fuse?
It's not, so our goal is slightly, I think, so for the super resolution, we just need to find the same cell, right? We don't have to pixel level registration. So that's a little easier. If it feels that pixel by pixel, it's a little harder.
Yeah, definitely. Thank you.
We are looking through the nuclear membrane to see the chromatin inside the nucleus. Doesn't the membrane itself or the proteins on the surface of the membrane block your view? How do you get beyond the membrane itself?
So these are, so this label is immunofluorescent label, right? So the lipid essentially is polymerized, otherwise the antibody won't get in. But the proteins we look at, they're nuclear membrane proteins, so they are located on the membrane, it doesn't block.
You're looking on the membrane of the nucleus itself.
So this sectioning. So the nuclear membrane itself, they are at the periphery, right? So when we image, we section the optical section that located inside. So we get like the rim of the nucleus, right? And sometimes if for cells, we actually we have projects specifically looking at nuclear membrane itself. In that case, you can go to the bottom level or top level, typically the bottom layer, just to look at nuclear membrane. But optical sectioning, you can go inside.
So you don't need any particular wavelengths to penetrate the...
No, these are microscopic, so the nucleus is probably 10 microns or something. It's not, it's the penetration is not an issue here. Yeah.
Is there any questions on there? Any other questions?
So, I thought you were covering your windstorm thing.
You, if I remember the steps correctly for that, you kind of did a deconvolution of your image by removing the background, then taking the Fourier transform, and then performing a linear deconvolution before doing the inverse Fourier transform.
Right.
And it seemed like from what you said, like you were doing these Fourier and inverse Fourier transforms in order to cause this linear deconvolution to like not be as noisy.
So the one critical step, I think, if you see the slides, we have a step that truncate the high frequency noise. That is critical. Let's see if I have that. I think this one, right? So this step here.
I
So
see.
that's so otherwise you will have higher. So this red area here, the reason it's red is very high value because these are really high frequency noise. So that part gets truncated, so that you end up getting this component.
So would you get the same results if you did like the inverse filtering in just like before the Fourier transform and then just doing a like low pass filter afterwards? Or do you have to do that inverse filtering in the Fourier domain?
In the Fourier domain.
Okay.
Yeah.
Why does the filtering have to be in the Fourier domain?
Because it's, well, one reason is, because you get, it's faster. Lots of these, when you do those filtering in the Fourier domain, it's linear operation, right?
Okay.
So it's faster. Yeah.
All right, that makes sense. Thank you very much.
Oh, sure.
Have you ever explored using a chromatic confocal microscopy to look at the depth, or does that not work with super resolution imaging?
So confocal, so people use spinning disk confocal to capture the blinking event. So that works. I think the trade-off is you need certain time to accumulate. So if the exposure time is too short, you end up with lower resolution. Because in single molecule localization base, the photon counts essentially determine your resolution. But people have done, especially for 3D, spin disk confocal is actually an ideal case because they remove all the background with the sectioning.
Any other questions?
I was just wondering, when you were talking about developing the mesoscale microscope with the larger objective, you showed that image of having a lot of field curvature around the edges, and you talked about trying to account for that or compromise for that. I was wondering what type of techniques you use to actually account for the curvature out at the edge of the field and how accurate that is.
So we didn't really go to and now I look back, there's probably a better way to do it. The way we did it, we took multiple steps and then we just refocus automatically find those regions. But I think there's either some wave form correction or some deep learning base that could be used to make it better. Because we do take, I think, 5 to 10 sections, stacks.
Any other questions, folks? I have a quick question. So for your mesoscale microscope, can you also do single vertical microscopy with it?
It's a great question. We actually had, so the challenge there is, it rely on the photon, the photon counts, right? So we had some idea of using some kind of photonic chip. Because the NA is low, the thing, it's not you just losing the resolution, you're losing the photon count. So that's really the biggest problem. So we were thinking of using some kind of photonic chip to do that. But we haven't figured out how to make those chips yet.
Is it 16-bit resolution right now?
Yeah.
Okay. Oh, okay, even that.
Any questions? If not, okay, let's take Dr. Yu again.